after the final, we just play backwards.
There we go. All right. Okay. Well, also, I want to also extend to you guys, any of you guys who don't have a place to go for Thanksgiving, you are welcome to come to join my family and I at our house. If you would like to put up with lots of screaming kids and all the fun that comes along with that. So just FYI, you don't feel obligated by any means, but I have had students over before, so if you guys would like to, you are welcome to. All right. Okay, so continuing on. So we talked about we were talking about P-type ATPases last time, and the one that we were talking about was the sodium-potassium pump. All right, so we went through this whole thing last time, talking just to review. So the way the P-type ATPases work is they have two different conformations. They have a conformation where they're not phosphorylated and a conformation where they are phosphorylated. They get phosphorylated in the spartate residue, like shown here. All right, and so the way they work again, just quick review, is in the non-phosphorylated state, they will bind to certain chemicals that they're going to be transporting. So in this case, they'll bind to 3 sodiums on the inside of the cell, on the inside part of the cell, all right, forming a complex with them. And what then happens is that the ATP that they can bind will get hydrolyzed, and the residue in the protein will get phosphorylated. Right? This now causes a conformational change where those 3 sodiums that were bound inside of the protein, now basically a door closes on the inside of the membrane of that protein, and a door opens on the outside. So now that they are free to diffuse to the outside of the membrane. Now, at the same time that this is happening, right, so now we have the phosphorylated E2 state. Right, the protein is now open to the outside of the membrane, and part of the reason why it released those sodiums is that the conformational change will also cause the binding sites for sodium to kind of go away and become better binding sites for potassium. All right, so now potassium is able to bind to that open side of the protein. Right. They'll come in there, bind to within the pore, right? And now the phosphorylated aspartate will get hydrolyzed. So this now reverts the protein back to its original conformation. So the door to the outside of the cell closes, and then the door to the inside opens up. All right. So now those potassiums are exposed to the inside of the cell where they can be released again. So the conformational changes that are occurring are both kind of opening one side of the opening access to one side of the membrane and closing access to the other, but they are also modulating the affinity of the inside of the pore for either sodium or potassium. All right, so basically, when it's open to the side where it wants to bind sodium, the binding sites are ideal for sodium. And when it opens to the other side, they're really poor for sodium, so it releases it, but they're ideal for potassium to bind. All right, so little side thing here. So, being able, a lot of these ion pumps, one of their main functions is to establish that electrochemical gradient across the cell membrane. All right, and that electrochemical gradient, what we saw when we were talking about the nerves, can be used basically to cause action potentials. Right? Basically by passive diffusion of the ions back into the cell. All right, so these pumps are really important for establishing the conditions that are needed for neurons to fire or muscle tissues to fire. Now, these are 2 medically relevant compounds right here. They're called digitalin and wabane. All right, these 2 chemicals in very high doses can be lethal. So like wabane is actually put on the tips of the darts by some native tribes in South America to kill animals when they're hunting, for instance. But at lower concentrations, right, these can be used to actually strengthen heart contractions. All right, the reason why is these compounds will basically bind to some of these P-type ATPases. So one specifically they'll bind to is the sodium-calcium pump. All right, so the sodium-calcium pump is part of what's contributing to the sodium gradient being high on the outside of the cell, so it tends to pump sodium out, along with the sodium-potassium pump that we just talked about. But it also pumps calcium to the inside of the cell. And calcium being inside the cell is very often a kind of an initiator of action potentials, right? So having a high concentration of calcium inside the cell leads to stronger muscle contractions. We're not going to go much more deep into it than that, but you can see that the point here is that by modulating and affecting how these pumps perform, we can basically potentially treat nervous problems, muscle problems, stuff like that. All right, so here's another type of channel, another example of a P-type ATPase. All right, so there are numerous types of P-type ATPases, and the same pattern, we'll see the same pattern over and over again with regardless of which one of these we're talking about. So for a P-type ATPase, we expect to have two different versions of the protein, two different conformations, an E1 conformation and an E2 that's phosphorylated. All right. One of the conformations, the E1 in this case, will bind to ATP. Right? It will bind the ions that are going to be transported. In this case, this particular pump is trying to pump calcium outside of the cell. Right? So it will bind calcium on that side to high affinity sites, basically, and the ATP will be used to phosphorylate a residue on the protein. Right? This causes a conformational change. The conformational change shuts the door on the previously open side of the membrane, opens the door on the other side, and it also weakens the binding site. So now the calcium has access to the other side of the membrane. The binding sites are weaker, so the calcium ends up being released on the other side of the membrane. In this particular example here, this calcium ATPase actually pumps two different things as well. So it pumps calcium out, or calcium into this, outside. I hate how this textbook kind of flips the membrane side. So it's pumping calcium out, but then it pumps protons into the cell. Right? So it's also involved in maintaining pH balance, stuff like that. Right? But again, the way it resets itself is the phosphate on the phosphorylated protein gets hydrolyzed off, and the subunit basically reverts to its original conformation. Okay. So this is, again, what it looks like. You can basically see that there are these transmembrane domains here that will form the pore. And then it has separate ATP binding domains inside the cell where the ATPase activity actually resides. All right. Now, the P-type ATPases are not our only example of ATP-dependent pumps. Right? We have another very major family of ATP-dependent pumps. They're called the ABC transporters. This is a huge family of transport proteins. All right. ABC transporters are often involved in transporting larger molecules. So, the P-type ATPases tend to pump ions, individual ions. These guys will tend to pump larger organic molecules. But there are some of them that do pump ions as well. So, the example shown here is a protein called multi-drug resistance protein. So, one of the ways that bacteria can become resistant to certain pharmaceuticals, certain antibiotics, is just simply by pumping them out of the cell. Right? And this particular protein is involved in that reaction. Basically, it will bind antibiotic compounds and pump them outside of the bacteria. Right? And as a result, that antibiotic is basically rendered ineffective. Right? So, all right, the way these end up working, all right, this is showing you, basically a transport of a certain this protein molecule here. Right? Basically, these proteins, again, they're similar to the similar in some ways to the P-type ATPases in that they use ATP to carry out this function. But in this case, the conformational change is induced by ATP binding. All right, so the yellow molecules down there, that's actually the ATP. All right, so when ATP binds to these proteins, basically a channel that was open on one side of the cell changes to a channel that's open on the other side of the cell. So initially, this pink protein can come in here into the channel, bind on one side of the membrane, then ATP binds, and all of a sudden the conformation changes that protein is now released to the other side of the cell, or other side of the membrane. Right. Now, these ABC transporters, they do hydrolyze ATP, right? But they use the hydrolysis of ATP to reset themselves. So when they're getting reset, the reaction that's happening, the ATP is just getting converted to ADP and phosphate, neither which bind. So these both basically leave the protein. So it gets hydrolyzed, they leave the protein, and the whole protein resets. So you can kind of think of the ATP hydrolysis here as sort of the reset mechanism in this particular case, but there's never a phosphorylated residue, at least not like we have in the P-type ATPases. So it's ATP binding itself causes conformational change. The hydrolysis resets the conformation to the original state. All right, one other thing I wanted to say here. Yes, so so one example here, is of an ABC transporter, as I said, is multi-drug resistance protein. These ABC transporters are also relevant to other human diseases. So, for instance, cystic fibrosis. It's a disease that basically causes thickening of mucus in patients, and they can end up, basically suffocating on their mucus and stuff like that. It's a really, really horrible disease. But the entire disease is caused by a mutation in one of these ABC transporters. And what that ABC transporter is pumping out of the cell is actually a chloride ion transporter. So basically the chloride balance inside and outside the cells gets screwed up. And this causes then thickening of the mucus in these patients, and they end up having some very serious health complications as a result of that. But just kind of illustrating for you in a different way the importance of these transporters. Right. Now, all right, that's it for the ATP-dependent transporters. Right. The next type of transporters we're going to talk about are ones that use chemical gradients to help do pumping of molecules. Right. So the example shown here. So we actually have a few different types of transporters here. All right, what we're looking at in this case, we're looking at the intestinal epithelium. All right, so inside of our intestines, some of the cells that are absorbing food from our diet, right, and how they're functioning. So, here's one of those cells blown up to the right. Right, the cell, it'll have the sodium-potassium pump to help maintain the gradient of ions inside and outside the cell. And so, again, this cell will have a high concentration of sodium on the outside, low concentration inside. Now, these cells have a really specialized function in that, the entire rest of the body can't get glucose from our diet unless these guys take it in and then shoot it out. Right? So they've got to both be able to do a good job of uptaking glucose, but then also transporting glucose into the bloodstream. All right, so they've got a really specialized function here. Now, the way that they end up taking in glucose, so you might imagine, inside of these cells, the glucose concentration is probably pretty high because they're continually absorbing glucose from the intestines, right? Assuming we just ate. All right, so how can we keep glucose going in? Well, the answer is that the glucose transport uses a symporter here. So symporter means that we have two different molecules moving in the same direction through a transporter. Now, the glucose basically we want to be able to pump glucose in even when the glucose levels in our gut are low and they're high already inside of the cell. Right? How can we achieve this? Well, the movement of sodium to the inside is favorable, right? So basically, there's already a high concentration of sodium on the outside, a low concentration inside. So the sodium really wants to enter the cell. Right? It's energetically favorable. We have a negative delta G for sodium to enter inside of the cell. Right? We have a positive delta G at times for glucose to enter inside of the cell. Right? What happens is that basically we're taking these two reactions, sodium outside moving to sodium inside, and glucose out moving to glucose in. All right, this, if we're looking at the delta G is here. All right, this one has a very negative delta G. Right, this one we'll just say has a positive delta G, a slightly positive delta G. All right, if we do both of these reactions at the same time as a part of a coupled chemical process, right, we basically add together the two reactions. All right, what ends up happening is the larger negative delta G of the sodium ends up canceling out the slightly positive delta G for the glucose movement, right, and we end up with an overall negative delta G. We can basically add together the two delta Gs. All right, so the way these type of proteins tend to work is they will have binding sites on the transporter for both sodium and glucose. All right, and once the sodium ions bind, whatever the favorable transport material is, once that material binds, in this case it's sodium, right, that's going to induce a conformational change in the protein to cause it now to open to the other side of the membrane. Right, and then in this case, both the sodium and the glucose will be released to the other side. Now, just to kind of gets into dietary stuff. All right, once glucose is inside of the cell, all right, glucose can then be passed into the bloodstream by passive transport because these intestinal epithelial cells, because they're continually importing glucose, it will tend to have a high concentration relative to the capillaries in the bloodstream. So because of this, glucose movement from the inside of the cells into the bloodstream is a passive diffusion process. All right, the import into the cells, the endothelial cells, is often an active transport process. Right now, this actually gets then into, for instance, the design of drinks like Gatorade. Right? So Gatorade, you can have or Powerade, whichever one you prefer, can have two different functions for an athlete. Right? It can both provide salts to their body, basically prevent cramping, and also provides energy. All right. Well, and also you'll see, for instance, in the medical profession, if you're dealing with a patient who's severely dehydrated, right? One of the things you will often do is actually give them some glucose along with their water, right? Glucose solutions and stuff like that. Now, why? Right, the reason why is because both sodium and glucose get imported into out of our stomach at the same time. So they're codependent on one another. So basically having a lot of glucose around, right, is going to tend to bring sodium along. Having a lot of sodium around will tend to bring glucose in. Right? And if we're bringing lots of sodium in, this tends to increase the salt concentration inside of here. What do you think is going to happen to water absorption by the cell? Water is going to rush in as well because we're increasing the salt concentration. All right, so this is why, for instance, kind of giving Gatorade and stuff like that, or a Gatorade-like solution can actually help with dehydration, right? It's because we're bringing in not just glucose, but also sodium, and water comes along for the ride as well, just to balance out the osmotic pressure. All right, there we go. All right, another example of this sort of thing. All right, we're kind of into using chemical gradients now to transport other molecules against their chemical gradient. So one example is the sodium and glucose transporter. All right. This example here is a protein called lactose permease. All right. The role of lactose permease is to move lactose from outside of a cell. So in this example, this lactose permease is actually a bacterial protein. So we're talking about a protein found in bacteria. I want to stress that. So the periplasm is the outside and cytoplasm in. They can't seem to keep their sides of the membrane straight.
You said this is in bacteria?
Yes, yeah. All right, so bacteria need to basically be able think about it from a bacterial standpoint. Bacteria floating in a lake or a pond or something like that. The environment around them is actually pretty devoid of sugars, right? Because there's a lot of competition for any available sugar. The concentration of them tends to be pretty low. So for a bacteria, they need to be able to basically concentrate food and pump food inside of the cell against a chemical gradient, because presumably the amount of food inside the cell is going to be higher than the amount outside. All right, so in this case, they're pumping lactose from the outside to the inside of the cell. All right, but along with this, all right, so it's let me back up. Lactose is basically being pumped against this chemical gradient, and so we have to either burn ATP to do this or use another chemical gradient to facilitate the process. In this case, we use the proton gradient that's created by bacteria. So bacteria do the same thing during metabolism that our mitochondria do. So basically, they pump protons outside of the cell as part of the electron transport chain and use this energy of that proton gradient to make ATP. Right, so bacteria and mitochondria can be very similar in this respect. Right? So basically, on the outside of the cell, in general, we have lots of protons. On the inside, we have a low concentration of protons. So it's very favorable for the protons to cross back inside of the membrane. Right? It's unfavorable for the lactose to cross. All right, so what ends up happening here is that we have the protein where it's open to the outside of the cell, and it's capable of binding both the protons and the lactose. Right? Once both are bound, this triggers a conformational change again that's going to open the protein to the inside of the cell, where it can now release both of these across. So because they're getting pumped together, we can basically add together the delta G's of the half reactions. So the delta G for the proton getting carried across is a very negative delta G. All right, the delta G for the lactose getting brought across is a slightly positive, so they add together and we get an overall negative delta G then. All right, once they've released on the inside of the cell, the protein is now just free to reset itself and have the binding site open to the outside of the cell. All right, this is just showing you the inside of the cell here. So you can see the binding site basically for lactose down in the bottom of this channel. Here in black, that's the lactose bound in there. And so you can it's pretty easy to imagine here actually how the top part here could close, and this then causes an opening of the bottom portion, right? These parts kind of move apart here. All right, that is it for this active transport case here. All right. A couple of things to add here. Right, so I want you to be able to distinguish between passive mediated transport. So passive transport always is going to have chemicals moving down their chemical or electrochemical, I should say, gradient. Active transport is always going to be moving things against their chemical gradient using some sort of energy input, often ATP. And secondary active transport is specifically that type of active transport that relies on a second chemical gradient to pump one chemical across. Right. Can somebody explain number 2 here? Explain why the sodium potassium ATPase and calcium ATPase only carry out transport in one direction. Like, so let's go back real quick. And see if you guys can kind of reiterate what I said in your own words. All right, this guy here, all right, the sodium pump. All right, so it can bind sodium and bring it outside of the cell. Why doesn't it just bind sodium on the outside of the cell? Because there is sodium there. There's actually a lot of sodium out here. Why doesn't it just bind sodium and bring it back in? No, so the delta G would actually be negative for that for sodium to go backwards, right? There's something that happens to the protein itself in the binding sites. So the conformational change does what specifically, though? You're on the right track. There's a conformational change involved. Which side of the membrane, let me ask you this, which side of the membrane binds sodium more tightly? Or which state of the, let me retract what I said or phrase it in a better way. Which state, the E1 or the E2 phosphorylated state, which one of them binds sodium more tightly? E1 binds it more tightly. So the amino acids in the channel and stuff are basically more optimally located and oriented to bind sodium more tightly in the E1 state. In the E2 state, presumably they'll probably move further apart because potassium is a larger atom, right? So they'll move further apart so they don't bind sodium very tightly anymore. They only bind potassium, right? So that's why they basically don't bring sodium back in. It's because the E2 state is just incapable of binding sodium, right? If it was very capable of binding sodium, then the sodium would just be brought right back in. So we have to have, in kind of the take home here is that for the P-type ATPases, we have to have a change in the affinity, change in the binding constants along with these conformational changes. So let me put it in a little bit of a review for the exam. Let's put it into KD terms. Right? Is the KD for sodium, which one, E1 or E2, which one has the higher KD value for sodium? Right. So I have a vote for E2. Everyone else agree, disagree? Which one has the higher KD? Yep. So the KD for sodium, we're thinking about the KD for sodium. I want to make sure to specify that. KD of Na plus, I don't know why I put a 2. KD for sodium for E2. So a high KD means weak binding, right? So for E2, it's going to have a high KD for sodium. E1 will have a low KD for sodium. All right, so just this is kind of, I guess, a way that you can start to synthesize stuff from different chapters and whatnot. So that would be a fair question on an exam, so just keep that in mind. Not in this next exam, but on the final exam, I mean. Just to also add to that. Oh, all right. Explain how an ABC transporter might function as a flipase. So actually, the flipase enzyme that we talk about in chapter 9 that you guys do have to know for this exam. All right. What is the role of a flipase, first of all? Yeah, basically it takes one phospholipid that's supposed to be on one side of the membrane and flips it to the other, right? So now flipases are actually another example of an ABC transport protein. All right. So how does an ABC transport protein work in general again?
Conformational changes when the ATP is bound to it.
Yeah, so basically initially the ABC transporter would probably bind that phospholipid, right? All right, then undergo a conformational change that's going to will bind ATP, and the ATP is going to cause a change in the conformation. It's going to flip that phospholipid to the other side of the membrane. And then how does the channel reset itself?
The ATP ATPs, or yeah,
Yeah, the ATP gets hydrolyzed, right, into ADP and phosphate. They both fly out of the active site, and now that protein is now ready to bind a phospholipid again and repeat this flipase activity. Right? Yeah.
So if the hydrolyzing of the ATP happens after the conformational change, it looks like I guess the energy from the breaking of the ATP-ADP bonds would just kind of be lost as heat, or what would happen to the energy from the?
Sorry, it's a good question. I have to think about this one for a second. So some of it's going to be lost as heat. That's always the case with any chemical reaction, but most of it's actually going to be, so the energy has to ultimately be put in towards the transport process, right? So it has to be used either to directly move something from one side to the other, or to cause this like a conformational change or something like that. So my guess, and I don't claim to know everything about every protein, sorry, is that the energy as the ATP is actually getting hydrolyzed, that it is actually probably the ATP binding is kind of causing a high energy state of the protein to a certain extent, and then the hydrolysis is basically letting that high energy state come back down. Is probably what's happening. I'd have to read a little bit more about it. I'm sorry. Yeah. So for any active transport, what is the ultimate source of free energy for secondary active transport? The gradient of some sort of a favorable gradient, right? So in the case, it could be the sodium gradient, right? Or like the proton gradient. There are other examples of gradients that can be used here. But basically, the ultimate source of that free energy change that makes an overall thing favorable is some sort of another gradient, electrochemical gradient. All right. That is actually it for chapter 10. I'm not going to start on chapter 11 for you guys today. Do you have any questions prior to your exam? Any question? And if you want me to, I can also give you guys put the images up for your gels, and we can talk through those, make your lab analysis really easy. You want to do that instead? All right, give me just a second. Gotta email these to myself here.
You could AirDrop if you use a Mac.
Yeah, if I had a Mac, right? Yes.
When there are two alcohols on the same side, that's when it's in the alpha form, right?
So it's in the alpha form when the alcohol is on the same side or on the opposite side as the CH2OH group. That's what you want to look at. You want to look at the CH2OH group and the